Thank you, Lee, for this spectacular talk. As always, we will I believe there are many questions from the audience. Probably you can ask Lee during the break. Or also, we will have a panel discussion session where you can ask questions. My name is Guangrong Qin. I'm a senior research scientist in Ilia's lab. It is my honor to introduce the next speaker, Dr. Sui Huang, who is a long-term friend with Ilia and our lab. So Dr. Huang is a professor at ISB here. He's a molecular biologist with strong background in theoretical biology. He received MD degree and PhD degree in Switzerland, studying complex systems and more specifically gene networks. This allowed him to bridge fundamental theory with translational research. Now that we are entering in the area of phenomics, he shared a long-term friendship with Ilya of more than 2 decades, and their love for Bach and Boolean networks has been, and they have been collaborating with each other for over 15 years. Today, Dr. Huang's talk will be about in front of memory of Ilya from Boolean networks to digital twins. Welcome.
Oh. All right.
Thank you very much, Wang Gong. And good morning, everybody. Thanks all for coming. And good morning to Marina and Vladimir and Anna. Yeah, it was Ilya left a big void because we shared the love for math and music. And one of us was just a professional, and he never made me feel that way. So there's some sadness because it's almost like the whole thing is reduced to one hand clapping. But also I am very privileged to have this very unique memories of Ilya, who will always be inspiration and illumination to the path forward. So his intellectual contributions are undescribable, and you see that. And that's important as we move forward and do what Lee just presented, uniting big data and AI and digital twins. So I was tasked to talk about the digital twins, and I haven't worked on that. So it will be a lot of discussion and ideas. But Ilya started 25 years ago with Boolean networks, and I will try to show you the path from Boolean networks to digital twins. I think Jim mentioned all the papers he's published. You can see a theme here emerging. Critical dynamics, probabilistic networks, multi-scale representation. Many authors are here coming from Finland. So very grateful for that. And you can see that over 20 years, he used a probabilistic Boolean network in one of his latest papers with Bahar, who study differentiation, transdifferentiation of cell plasticity, which is now a big thing in cancer biology. So, but there's one gem that I think we forget, that's this paper here in the middle, hidden. I think Jim mentioned that. That's this year. We liked it. It was 20 years ago when I read it. Physical review letter, so a prestigious journal in physics. And here, with Boolean networks, we mean, of course, a gene regulatory network. And I'll try to unpack the theory for you in a few slides to see whether, because I really think it's important. So here in Ilya deals with gene regulatory networks, and that's most of you are familiar with that display with a network of genes regulating each other. Now, Ilya introduced two concepts that the activity of a node in an abstract network is a gene, and then sensitivity of a Boolean function. The Boolean function is the promoter region. It tells you how all the genes that control that use logics to determine whether that gene will be active or not. And it can be more sensitive or not. And there's a mathematical, the Boolean network. There's a mathematical description, actually pretty straightforward, but nonetheless brilliant. And I'll just tell you the conclusion of what happens if you look at sensitivity, especially the average sensitivity of all the Boolean functions of all the genes in a network. And this is the quote from the paper. So he says, so we have an analytical method that allows us to determine whether a specific network is ordered, chaotic, without having to run computer simulations. So that's the point. It's purely analytic solution. As you know, in math, the analytic solution is when you have the highest goal you want to achieve and not these dumb computer simulations. And then he says, given a concrete network, and I will like the term concrete, so it's not theory, it's a very specific network where you have a wiring diagram, we can compute the average sensitivity for each function, so each promoter logic, and average them and obtain a single number that would tell you in which regime the network operates. Now we have to understand what is the regime in which networks operate. That is dynamical behavioral regimes, going back to Stuart Kauffman 50 years ago, right? And we can illustrate that here. So a network, a gene network, controls how a state, so these green balls will always be state. A state is a state vector, so gene 1, gene 2, gene 3, and so on, and all have the values with a row in your Excel spreadsheet. That's a state. And that moves from one to another, but since genes are connected to each other, they have to follow a particular path in a change. So every change in complex systems is highly constrained by the network. In this case, we have the regulatory network. The green line is called a trajectory. And if you now look at the sensitivity and plot all possible states and where they go, you see pictures like that. These are the three regimes that now are predicted by the value of Ilya's average sensitivity. Low sensitivity, all the states will collapse into one stable state. If you have around 1, it's what Stu and others, pioneers of complex systems, call the age of chaos. And that has interesting patterns. And if the number is high, with chaotic behavior. So the yellow one are the steady states. So one of those states that don't change anymore. And if all the trajectory move into them, then you have an attractor state. That's just background definition. And now it's obvious that these patterns can be represented in a different way, which is shown here. Again, the state of this network with respect to the values of the genes is a given state, it's a given vector, and we can now map this in the landscape to capture the gravitation towards a state. And that's why we have these attractor landscapes. And in this case, you have just one attractor. In here, you have multiple stable states, you have multiple valleys. And then in the chaotic way, you have just a flat landscape with very shallow attractors. And so the slope will indicate that there's a drive towards the stable states. And then we have a state, a cell in that state. So every ball is a state of the network equivalent to a state, right? To a cell phenotype. In the chaotic regime, the state would just aimlessly migrate through the entire state space and exploring all possible combinations of gene activities. So what is neat is that in this age of chaos, you have these multiple states. So you have a compartmentalization of all initial states towards a relatively few number of stable states. And therefore, the system can capture information and process information. And it was the whole idea going back to Waddington, Max Delbruck, actually, and then Stu, that these attractor states are cell types that we see. So in Ilya theory, it allows us to look at all the promoters and the logics we don't have, but maybe one day soon, and then predict whether a network will be in which regime. And it turns out that most living systems will operate in that age of chaos regime. Thanks to AI, I can now draw this landscape in 3D in a much more complex way. So in reality, you have many more valleys. And the interesting takeaway now, which will be important for digital twins, is that a given wiring diagram, and I will show you they are different for you and you and you, will map into a particular topography of the landscape. And that is how you move from genome to phenome and can predict a phenome by looking at the genome. And as Lee mentioned, that's our goal in future medicine. So this is just to remind you of the basic biology. So we have networks of genes come from the fact that some genes are transcription factor acting on other genes and activating them. The center figure is abstraction that go back to, I think, Eric Davidson, also at Caltech. And just to remind you, what we measure is activity of every gene in transcriptome, for example. So we see these vectors of values, and for every cell type of another gene expression profile. And to remind you, every profile is a state that maps to a position on the landscape. And Waddington intuitively, of course, proposed that. But now we have developed theories to formally show that it's possible to define a sort of a quasi-potential. It's not the energy potential. It's something else, but it gives you the a notion of a driving force towards valleys. And as you can see now, it turns out that if you write down all the equations, you will get this landscape, but many attractors are never used. This is a mathematical byproduct of a complex system. They're never used by the system, and Stu noticed that early on in the 70s. So we have attractors state here. Sometimes cell can go into that unused regime of manifestation of combinations of activities, of gene regulatory interactions and gene activities. And these unused states, some of them are also stable, but they were never seen by evolution. They were never optimized and made to serve the entire organism. So if a cell goes there, in these unused regions, there could be cancer cells. And I think that's the formal prediction of the very existence of cancer that Sue proposed in this 1973 paper, I think. And then we start to build on that, Ilya and Sue and myself, and try to understand cancer in much more fundamental ways. Now, digital twins, there's a lot of definitions I know, and many people have different notions. And of course, Ilya was a good partner to fight about what it actually means. Here is my functional definition. So X is a person, X prime is your digital twin. Now, what it is not. I know people don't like negative definitions, but it's important because it's not just a mode in silico mode. We have had that since the 1980s, with IBM Watson, CyberMouse, virtual humans, and all these things, right? It's not just a computational model. There's one aspect that has to be there, and that is the twin aspect, and that is that a twin X prime is an in-secular copy of the original one, and it has to recapitulate all the individual idiosyncratic features. So it's not a model of a class of organisms. So you just model a tumor, and you think it's just a generic tumor. That's not a twin, that's just a model. It has to be individualized, and what goes in, what is individual, right? That is the genome and your past experience. So therefore, digital twins are at the heart of personalized medicine. So just let me remind you of what it meant in engineering, because before biologists butchered that term. A digital twin, for example, has been used for jet engine. So every jet engine is identical to some reasonable extent. But if you build them into planes, they undergo different histories. Some might fly on the North Pole routes and so to exposed to different environment. Starting from the identical design, we have the blueprint, which is identical. The blueprint of every jet engine is identical, but the history is not. And the blueprint of human genome, they're not the same, and that's a problem. So in the case of engineering, we could keep measuring data in real time, and now put them into this equation of this wiring diagram that you know, and it's universal. That's the digital twin. And with that, you can then predict what's happened to that engine, given the different maintenance history, exposure to different conditions. And then with that, you can predict the next step. What will happen with this engine? Will it fail in the next year? Does it need maintenance? That's what people mean by digital twins, strictly. So you need a blueprint that is universal, and you have individual histories. Now, can we do that for humans? We're not identical. People are different. we have genetic diversity. Each person has its own genome and therefore its own wiring diagram and therefore its own landscape and its own phenotype. These are the genetic variants that we measure. And so what can we do with that? Humans are also exposed to environmental conditions that differ, but they start with already different genotype. And that gives us the opportunity to combine a given genotype and a phenotype that's different and exposure to the exposome or environment. So we have this problem here that people are different, and we have the nature and nurture issue. Nature, your genes, nurture what you experience. And how will that affect health trajectory? So the problems here of individuality of humans, we are not clones, therefore we don't have a blueprint that we can use to model things. And as Jim also said, our wiring diagram, what we know is vastly, vastly underdetermined. It's just not realistic to do the same thing that we could do with jet engines or nuclear plants or whatever people use digital twins for. And maybe that's the reason why we need to go back to big data and just sometimes do it with data and without model. That's a new trend, especially now with AI, just stitch mechanism. We have to be open to that. And I know many people are modeling things. It's fine, I like it, but we don't know what's the future. So it would look like that if you have human digital twins. This is just my idealistic model. You have S, which is real world data. D is your digital data, and you have a model, the probability to go to a next stage. And that could be where you have your mechanistic model. You make measurement, you inform your internal digital state, which is again a state vector. Many variables are never measured, and then you can predict the intervention, and that leads to a response, and then you feed back, and you have this loop, and that's the idea of digital. But we also have the huge cohort of people who are slightly different from each other. That's what the engineers don't have. And that's also a source of information. So UK Biobank, all of us, and FinGen, there are people, they all have slightly different gene wiring diagram, and that is a source that you can learn from. So a model would be ODE, mechanistic model. These are dynamical systems modeling. I've seen many faces, familiar faces, people working on that field. What is new is also knowledge graphs, where we put in all possible relationships between genes, proteins, metabolites in. That could be also part of the model, and or inform directly the updating of your digital twin. And now we have big data, as I mentioned. You can enter through AI and graph neural network and something this is just a training of the weights of the network, also enter in your digital model. And you also is a source of information for the knowledge graphs that I think some of us are involved in building. But as I said, then there's this field of data scientists that don't care about this. And they use dynamic Bayesian networks just based on states and probabilities of what's the most likely next state, and now AI and deep learning to learn a latent space on which similar things will be in the same neighborhood and update to the same state, which they wouldn't in the real data space. That's what AI does. And so just an example of existing mechanistic model, and over many years with Ilya, we all think of what's the right level of granularity that we need to do to the modeling. Maybe we don't need model the mitochondria, but just liver function. Then we have knowledge graphs, and as I said, we have at ISB many people involved in several big consortia building these huge networks of all possible biomedical relationships. And now what is neat is some of these nodes in these knowledge graphs are your genetic variants, all the polygenic risk score that Lee was talking about. They are genes, and typically they are in the regulatory regions of genes, and that is exactly the Boolean function that Ilya was studying, and they affect the dynamics, and they can also be entered in your model. So you have generic knowledge from big cohorts, like which things are correlated in a big cohort, but you have also or individual level idiosyncrasies like the SNPs or gene variants that individuals carry, they could also in principle be entered into your model, and then you have an individual level model prediction. But that's all dreaming. We can be much more simpler and take what we have. So just remind you of the theory of networks. I mentioned earlier, every network maps into its own landscape, and that goes back to this work by Ilya, purely theoretical, but I think it's correct. And now that we have sequences of all the promoter regions and SNPs there, we can really start to predict how a landscape would look like. And you can see the landscape will be different. That would translate into different propensities of cells moving in one direction or another, and maybe a higher propensity for certain landscapes for cells to move into these bad regions that have these cancer attractors. So that's how a propensity in a SNP manifests itself in a disease. And we have the analytical theory behind it. So coming back to this, I'd like to move to data now for the last slide. And as you know, in July 2019, Ilya was diagnosed with AML. And our reaction was, as scientists, to form a team to do everything we can. So it was called FOI, Friends of Ilya, which is foi in French, with face. And we did molecular profiling. The goal was to do everything we can and leave no stone unturned to see whether there is something there that we can help. Is that a digital twin already? And that's a question. I'd like to thank everybody, many people that have been involved in making it possible, helping, and spending the free time on it. One example, so what we don't have is a model. You just, I know, Guanro, you have an AML digital twin, but that's a generic one, right? So we don't have a concrete model of an individual. We have PubMed. We rely on existing mechanistic knowledge. We have knowledge graph, and we have a lot of data from TCGA. So we know a little bit about disease from looking at big cohorts. And then we have the individualized longitudinal multi-omics data. That's what we have. That's the status now of digital twin on a very concrete example. And Ilya was always so gracious and brave in actually participating in discussing his data. And I'm sure it will be thrilled. It has always been when we share data. So I'm just sharing it, even if there might be privacy concerns. But this is, we're friends here, and this is about the science. So that's a time course, 3 points. We took, as an example, single cell analysis from bone marrow. And we then realized we faced this new situation because he got a bone marrow transplantation. And suddenly we have 2 genomes in one person. So we have 2 genomes and 1 phenome. There's a unique opportunity to understand exactly what we want to study, how the genes and the difference affect the phenotype. And they're in the same body. So it's there's no batch effects here for the first time. So the idea was that young 19-year-old lady who gave bone marrow might have a healthy genome lacking the mutations that makes it much easier for cells to differentiate, and the mutated cells would have a much harder time, but much easier time to move to the stem cell attractors, the cancer attractors. So yeah, here is the data. So most of you are familiar with that. Every dot is a cell. This is the landscape just squeezed into 2D, and we can, based on markers, determine which cells are where. The circle are the blood progenitors. And now these are cells from Ilya's bone marrow. And this was from a later time point after transplant. So you see a lot of cells from the host. You can end from the graph. The graph is purple, and I made the dots smaller. And this is thanks to Michael's work to identify, and also Dave was involved and others, use the expressed genetic variants to identify which cell came from whom. And you can see that here, 95% of the cells in bone marrow were from the donor, which is good. That's what you want. So it's a chimeric. You still have a host cells, and these are the orange one. They just made bigger for optical reasons. If you look at them in this cluster of blood progenitors, you can see that the grafted cells and the host cells are different. So positions map into different state vectors, right? So they're different. They're not exactly the same. If you look at the cell cycles, you can do that. Host cells and the graft cells are similar cell cycles, so not one is out-competing the other, also good. So there are much more host cells in the home, but what we realize in that compartment, the red circles, the immature stem-like cells, there are many more cells relatively from the host than from the graft, and they are different qualitatively. Now, if you look at the host cells, ilia cells, overall, and they are just accentuated by making larger dots, you can see that these cells contribute to every lineage. They're normal to differentiate. They go to erythroid, they go to megakaryocyte, they go to T cells, they go to NK cells, they go to DC and macrophages. They look like normal. So I think the trick is that it's just a propensity of differentiation, which is reduced by maybe 10, 20%. That's enough to give you leukemia. And you don't see that by, gene sequencing, right? So the devil is really in the quantitative details. So if you look at another statistics, well, here, yeah, this is to show you how. So it's a minute difference in the propensity to differentiate. You could, if we had enough cell numbers, try to establish that ilia cells are maybe 10, 20% less prone to differentiate and more prone to enter that hidden attractor states that we normally don't enter. And we can also look at some mutations. These are well-characterized mutations. No one wrong, you know them. They are associated with AML, and they are luckily in the coding region, so we capture them. Cells that have mutations, are they enriched in the stem cell compartment? That's a question we don't know because we don't have enough numbers, but qualitatively, they can still differentiate into normal cells. That's the point. So cancer is not a qualitative disease, or so it appears like, but at the core, it's a quantitative disease, the shift of propensities to differentiate. And pathologists always known that they call cancer essentially a differentiation arrest. So only a small fraction of host cells are mutated. That's also interesting. So many cells don't carry these key mutations. And mutated cells can differentiate. And what matters is the ratio of differentiated versus stem-like cells. So I think that shows you that theory matters, a rigorous formal theory, and it might be helpful for digital twins. And underneath that, our ability to one day use the SNPs in the promoter regions to predict the landscape. It goes back to Ilya's work here. And so with all these good memories, I think we're also inspired by Ilya's contribution to look forward. And I think I'll thank Ilya for laying the foundation for future medicine. You have different genomes here. And thank you, everybody.